I worked in rare diseases when I was at the Broad Institute in Rare Kidney Disease, and really appreciated when you can identify the mutations and go after a specific population, how you could have more therapeutic success in being able to treat that. And so when the opportunity came to interview at Third Rock and interview at Fulcrum, I was using my history of rare disease to help evaluate if I thought they had a good opportunity. And it was very clear we had a good opportunity, especially with FSHD, because the mutation in the patients was just becoming clear. There was success in the muscular dystrophy arena. And I chose to work there in part because of FSHD at Fulcrum, but I also chose to work there because it was just a great group of scientists, maybe some of the best that I had met. And I thought it was a group that could be successful in this space that was well defined. There are really reasons to be optimistic. I'm an optimistic person in general, but I think there are reasons to be optimistic in that we as a therapeutic community it's generated so many ways to knock down a protein when we understand the protein to be deleterious to a function of a muscle or function of any organ. And so we have many possible approaches to try. Our challenge is going to be to really deliver it efficiently across a broad range of muscle types, because we know FSHD affects almost every muscle in the body, and that remains to be the challenge that we have to demonstrate to ourselves that we can do. But I do think the technologies for knocking down, toxic gain of function proteins are so excellent and have so many different avenues, that I anticipate 1, 2, or 3 may work. And then we get to evaluate how well to deliver and how efficiently that we can deliver each of those different approaches to muscle. The real challenges right now are that the society's role is to catalyze excellent science, to catalyze excellent clinical care delivery, to catalyze ability for patients to get diagnosed, and to catalyze education of neurologists who see patients. And I see the society working on all fronts. And I've had a real ability to be impactful already in the IRC that we ran this past year in Denver. We brought together the best scientists and excellent scientists from around the world, but we also saw some groundbreaking novel therapeutics that are starting to get described that are apparently impacting the cause of the disease. And I'm very excited about that. I value that the society works with every industry or biotech partner who's interested in working, and we work without reservation, and we work without bias to facilitate everyone's success. I also feel that the society is leading in thinking about what's required for patients going forward as we get to 2025. If a molecule is successful and is approved by the regulatory agencies, we're positioning ourselves to be in a position to help patients access the new medicines, get diagnosed for the medicines, and to help neurologists identify patients with FSHD. The other things is we're recognizing gaps in the area. So you or other members of the society have brought forward that we may have access to patient tissues, and we could generate cell lines. We could generate myoblasts that could be valuable for scientists. So there are reagents and FSHD specific tools that are not easy to acquire. Patient biopsies, patient surgeries help provide those kinds of things. And if we can identify creative ways to get those into the hands of researchers, we can facilitate the next steps. And then finally, I think the society has the opportunity to be creative in ways that biotech can't. If we bring forward new diagnostics, new biomarkers. We've done work in that area. It's a very challenging area. We've had some successes and some failures, but you have to continue to work to try to find the best possible solutions. And the society has the opportunity to be creative in a way that sometimes biotechs don't. And so that is a real pleasure as well. It means a lot, and I know the community well. So working at Fulcrum for those 5 years, I had the opportunity to see the generosity of patients who were willing to be involved in non-therapeutic biomarker-driven natural history studies. And those studies allowed us to characterize the disease. They allowed us to understand what challenges patients have, and they allowed us to generate really valuable tools that can be taken into the clinic. You know, they gave us muscle biopsies, they did whole body MRIs. And what I realized is that FSHD is a really challenging and very difficult disease that gradually steals from people their independence, their ability to move. And for them to still be as generous with their time, with their muscle tissues, with their with a challenging situation that they have, still to get themselves to MRI machines, still to be involved in clinical trials, I became very empathetic and compassionate for FSHD patients, and also very grateful. And we had the opportunity to learn a lot. And in that time, I realized that as much as we learned, we didn't have the opportunity to get that information all into the public domain. It just takes so much time, it takes so much effort. So if I could bring components of that knowledge, components of our learnings back to the patients, back to the society, and back to other therapeutic biotechs and pharmas, that I would like to be able to do that. And so being the chief scientific officer of the society entirely gives me the opportunity to give back